Hey, good morning, everyone. Before we started the lecture today, I wanted to kind of use this as another opportunity to sort of plug our molecular cell biology Bachelor of Science program here. I think this is the point in the semester where all of you now have a very good sense about what MCB is about. And I know that registration for fall classes is right around the corner. So I think this is a good opportunity to kind of remind you again that we do have a BS program in MCB. Now, I think most of you who are, life science students, you have maybe a broad sense of interests in biology. I know some of you are interested in becoming researchers in biology. Your interests in ecology, environmental sciences, things like that. Many of you are interested in marine biology, so you're part of our marine biology program. Many of you are interested in molecular biotechnology, so you might be part of MBBE. Right? And some of you are already MCB majors. But I wanted to remind you that, as you progress through your degree path, some of you, I think, are interested in pursuing careers in professional healthcare medicine, for example, becoming MDs or PAs, nurses, or other types of professional healthcare providers. And so for those of you, I think that, you might be thinking about things like MCAT, and you may be, for example, a biology major right now, bio BS, and that's fine, right? Because obviously you can be a biology major, a MBBE major, microbiology major, or you can even be an English major and still go to medical school. So what I want you to think about is it's not necessarily just about does your degree let you take the MCAT and move on? It should also be about which type of degree really sets you up for success once you move on to the next sort of chapter in your education. For example, medical school, PA school, dental school, etc. So this is a promotional poster that I had made a couple years ago that sort of summarizes what the MCB program is about. And importantly, I had the opportunity to invite some of my former MCB BS students who had graduated to kind of reflect upon how the MCB program and being an MCB major really ended up helping them as they continued on with grad school and med school. Okay, so I've put a PDF of this in Laulima under the main sort of resources file. So for those of you who may be interested in kind of thinking about this, I want to point out that it's not too late, right? This spring, the class that you're taking right now, Bio 275, is the first time that we've offered Bio 275 in the spring for several years. And that means some of you are in a good position as you are wrapping up, for example, sophomore year right now and looking ahead towards junior year to be able to think about, is this a good option to switch over to MCB? So I won't say too much about this right now more because I think this really only kind of is relevant to a subset of the class who are interested in these types of careers. But if you are interested, take a look at this poster. Feel free to reach out to me. I know registration for fall is just around the corner. So it's a good time to talk to me and to talk to a NatSci advisor if this is something that interests you and you want to find out more. Okay, so look for this poster in Laulima. All right, let's go ahead and start in with our lecture today. So today we're going to talk about the electron transport chain and how that links to ATP synthesis. So this would sort of end our discussion about the aerobic respiration that cells do to get as much energy as possible out of a molecule of glucose. Let's summarize where we're at now. So starting from a couple lectures ago, we began talking about how All eukaryotic cells, and certainly most prokaryotic cells on Earth, right, start processing glucose inside of them as the raw fuel source for energy. Right? They start by using anaerobic means that don't involve necessarily the availability of O2, of oxygen. Quick reminder, life evolved on Earth billions of years ago in an anaerobic environment, right? There was no atmospheric oxygen. So as a result, early life had to come up with glycolysis, an anaerobic means of kind of breaking down glucose to be able to get a couple of ATPs out of it. Remember, if glucose is the raw material, the raw fuel source, glucose has to be converted. The energy in its carbon-carbon bonds in the glucose has to be converted into ATP in order for the ATP to be able to be distributed throughout the cell so that all the different biochemical pathways, like all those enzymes that use ATP, right, can kind of plug into it that way. Right? So glycolysis, the splitting of glucose and the processing of glucose with some limited oxidation going on, gives you 2 molecules of pyruvate as the end of glycolysis. Right? From that, you end up getting 2 molecules of ATP, but you end up filling 2 of these NAD plus baskets that are the coenzymes that carry electrons away from an enzymatic reaction. Remember, enzymes oftentimes use coenzymes, right, as the source to transfer electrons to if they're taking electrons off of some other substrate. Right? And in this case, NAD plus, nicotinamide adenine dinucleotide, is a very good electron acceptor. Right? It serves as kind of this empty basket. I represent it as an empty basket in the cartoon. Once it gets a couple of electrons along with a couple of the protons that come with those electrons, right? Because remember, electrons aren't just going to travel freely as electrons by themselves. They usually have a proton attached to them, and therefore they're essentially a hydrogen atom, right? The empty the basket, the NAD plus, gets filled and it becomes NADH. Right? So from glycolysis, you end up, since you're making 2 pyruvates, each one of these pathways fills 1 basket. So you get 2 NADHs carrying pairs of electrons away. We learned last time that as life evolved on Earth, right, over maybe the first billion or so years of microbes doing photosynthesis to make glucose out of carbon dioxide and water, they are releasing O2 as a waste product. Right? When you take CO2 and H2O, put them together, and you reconfigure them into glucose, you're left over with an O2 molecule. And that just kind of bubbled up and became part of the atmosphere. So as the available amount of O2 in the atmosphere began to rise, now you have an opportunity, right, to use that oxygen to get more mileage out of your glucose molecule. So you're not stuck just making 2 pyruvates and then that's the end of what you can do with the glucose. Now with available oxygen, you can do more oxidation of those pyruvates, right, to get more energy out of the original glucose and convert those into more ATPs. We learned last time that the first step of aerobic respiration is the conversion of pyruvate that comes out of glycolysis, right? You oxidize the pyruvate and you make acetyl-CoA. And that step allows you to fill 2 more NAD baskets, NAD plus baskets. So you end up with 2 more NADHs. Right? Then the acetyl-CoA enters into the citric acid cycle. And it joins up with oxaloacetate and becomes citrate, et cetera, ketoglutarate, and it goes around succinyl-CoA all the way back to becoming oxaloacetate again. And along the way, those carbons that were originally part of the glucose become part of these intermediate molecules, and they systematically then get taken off, right? And as they're being taken off by different enzymes that work on these different intermediates in the citric acid cycle, you are filling more of these baskets. So you end up filling 6 additional NADHs and you get 2 of these FADH2 baskets. So I emphasized last time that the citric acid cycle itself, even though you do produce 1 molecule of ATP per turn of the cycle, that's not really what the citric acid cycle is about. The citric acid cycle evolved as a way of getting lots more of these free electrons taken off, right, of these carbon-carbon bonds. That's what really represents the carbon-carbon bonds in the sugars being broken, right, and the energy in the form of electrons being carried away by these baskets. So now you've made many of these filled electron-carrying baskets. What we're going to talk about today is what happens next, right? So all of these electron-carrying coenzymes, the NADHs, the FADH2s, they then file into the electron transport chain, which is step 4, and they empty these electrons into this chain. The chain is a series of large protein complexes that are arranged in the inner membrane of the mitochondria. And as electrons are flowing through these series of protein complexes, it makes these protein complexes pump protons, right? So this is active transport of protons across the 2 spaces inside of the mitochondria, right? Building up a very, very strong proton gradient, an H+ gradient. And then ultimately, what we'll finish talking about today is you will have certain other proteins as part of the membrane that will then allow those protons to flow back downhill, right? Because when you set up a really strong gradient, like 100 times stronger H+ on one side than the other, then the moment you have little channels that will allow them to flow downhill, right? You better believe that's a wonderful source of energy to tap into, right? And so these ATP synthase proteins do that. They will allow the protons to flow back downhill, and in the process, just like a crowd of people crowding through a bunch of turnstiles, right? I showed you a photo of that last time, right? Turning the turnstiles to get back in, right? You can harness that energy to do work. And in this case, the turning of the turnstiles will be the high throughput manufacturing of many molecules of ATP. So at the end of today's lecture, we'll kind of tally this up and just see how much ATP, additional ATP, does these steps 2, 3, 4, and 5, right, add on to the 2 molecules of ATP that you got just from glycolysis. Okay. One thing I did want to show you, let me back up a moment here. I mentioned that, we looked at the structure of nicotinamide adenine dinucleotide previously, right? So it's a dinucleotide. The bottom nucleotide is an adenine nucleotide, and it's linked through a phosphate bond to this other nicotinamide nucleotide on top. Right? And this nicotinamide group, it's nicotine basically, right? It's a good electron acceptor. So it'll happily take an electron and a proton on it, right? So what is this FADH2, right? It is another dinucleotide, but in this case, instead of having a nicotinamide nucleotide on top, it has what we call a flavin a group. So let's look at the structure of that, right? So you can see here, this is FAD. So it is a dinucleotide, right? The bottom part is obviously a nucleotide down here. The upper part, it's sort of less obvious because you can see here the sugar, instead of being a ring, right, it's sort of linearized, but you have a nitrogenous base up here, even though it's kind of this crazy one that's like 3 rings, right? So this is the riboflavin, and this is the oxidized form of the riboflavin. These 2 red arrows here indicate where electrons can be added, onto these nitrogens in the riboflavin, right? So if you add 2 hydrogens here, remember this essentially means what? 2 pairs of a proton with an electron, okay? Then this empty basket over here, right, gets filled with an electron or 2 electrons, right, and 2 protons. You can see here are the electrons, these are the bonds here, and then here are the 2 protons, right? So this would be the reduced form of riboflavin, right? So this would be the form, the FADH2, that is now carrying the electrons away. Notice, right, this structure is quite a bit different than the nicotinamide structure we saw in the previous slide. You can always go back and look at that. What that means is where electrons are being carried on these nitrogenous bases, right? Those electrons, the orbits that they're in, are in different energy levels, right? So the electrons that are here as part of the two hydrogens that are added on here, right? Each one of these electron orbitals is a different energy level than the electron that would be in the nicotinamide group being carried by the NADH basket, right? So in fact, the NADH basket, where it carries its electron, that energy, the potential energy there, is higher than the electrons being carried here as part of the FADH2. What does that mean? That means when it comes time to empty these electrons off, right, the amount of energy represented by the electrons that come off of the NADH basket is more energy than the electrons that are coming off of this FADH2 basket. Right. Oh, and I wanted to remind you, riboflavin, you're probably like, I've heard of riboflavin. What is that, right? Riboflavin, right? It's vitamin B2. Remember how we discussed the fact that, hey, we're always aware that vitamins are an important part of a healthy diet. We've known that ever since we were little kids, right? When I was a little kid, my parents used to give me these little Flintstone themed vitamins. I don't even know if you guys know who the Flintstones are. They were like a cartoon back in the 70s, right? Caveman family. Anyhow, so ever since I was a little kid, I would chew on these sugary vitamins, right? But you don't really think about what a vitamin does. And so I told you before, vitamins are often these electron carrying coenzymes, right? Like riboflavin, right? Or vitamin A, for example, right? We looked at vitamin A before, right? And so now you understand why they're important for normal health, because as long as you have plenty of these electron carrying baskets, right, all of these complicated enzymatic processes that are taking place inside of your healthy cell, right, are supported, right, because these coenzymes are helping the big enzymes actually do their function. All right, so let's go back to this cartoon, the cartoon that I like a lot, because it's a nice summary, right, of all these things that we've been discussing. So again, the 5 steps of cellular respiration that we're talking about here. Step 1 happens anaerobically. That's glycolysis. It happens in the cytoplasm of the cell. Then the pyruvate, that's the end product of glycolysis, is transported into the mitochondria. Step 2, pyruvate oxidation converts pyruvate into acetyl-CoA. Acetyl-CoA then feeds into step 3, the citric acid cycle, where one by one, the carbon-carbon bonds are being broken off, right, in order to then fill these purple electron-carrying baskets, right, whether you're talking about NADHs or the lower energy FADH2s, right, that come off the citric acid cycle. Then these red arrows then feed into step 4, the electron transport chain, which is what we will talk about next. One thing I wanted to point out to you, which I think is one of the most sort of, as you go on and learn more about science and biological sciences and cell biology, one thing to look forward to there are certain moments along the way where you have kind of this like aha sort of moment, right, where suddenly you understand something on a level that maybe you were aware of before, of this thing, but you never really thought about or fully understood how to make the connection of why it is that way. I think one of the most fundamental things as a biology student that we often think about is the fact that ever since you were a little kid, right, we breathe in air to get the oxygen and we breathe out sort of the spent air, when we think about that, because we're breathing out the carbon dioxide, right? So, ever since you were little, you breathe in O2, you breathe out CO2. But the connection between that O2 and the CO2, maybe you never fully realized or appreciated, right? Maybe you just thought somehow the O's and the O2 get taken apart and they become the O2s get attached to carbon and become CO2 and you breathe it out, right? The reality is, look at this now. This cartoon is wonderful because it shows you where the carbon dioxide comes from and where the O2 comes into play, right? Because along the way, the carbon dioxides are coming out as you are oxidizing intermediates like the pyruvate being turned to acetyl-CoA, and as you are oxidizing the different intermediates of the citric acid cycle, the breaking of the carbon-carbon bonds to break off CO2s from those big intermediate molecules. This is the CO2 that then diffuses into your blood, gets picked up by your red blood cells, and is transported back in your lungs to be exhaled, right? Where does the oxygen come into play? Right? This oxygen that we've been talking about as being the important driving force that allows this aerobic respiration to happen. Look where it is here. It's here. Right? It is at the very end of this next step, this step for the electron transport chain. Right? The oxygen is what finally receives the electrons that have been emptied by these electron carrying coenzymes and that have now flowed through this series of protein complexes that make the electron transport system, or ETS. And at the very end, those electrons are picked up by oxygen, right, to form water. Okay. And so when you think about that, right, you're like, that's kind of anticlimactic, right? I thought the whole point was we had talked about you take a glucose molecule, if you burn it with a flame in the presence of atmospheric oxygen, right, you have this violent exothermic reaction, right, the burning of a marshmallow. And we said, yeah, that's because as you disrupt all the carbon-carbon bonds in the sugar of the marshmallow, those electrons suddenly are looking around for something better to bind with, and you have lots of oxygen floating around. The oxygen is very greedy for those electrons, right? So it'll happily take those electrons, and that's why the sugar then burns, and you have so much energy released. It's flame, right? Without the oxygen, you're not going to have electrons being able to go somewhere better, and as a result, you're not going to get combustion, right? So we said in our cells, right, we don't do it that way. We do it through these very carefully orchestrated enzymatic steps of carefully breaking one carbon bond, try to get some energy out of it, break another carbon bond with a different enzyme, etc., right? And you're like, well, wait, I don't get it. But I thought the whole thing was, where's the oxygen? The oxygen is here at the very end, after all of that energy has been taken out in the form of electrons and is used to power the proton pumping of this electron transport system, right? The electrons at the very end need somewhere to go, right? And that's where the oxygen fits into the story. The oxygen is the greedy electron acceptors that happily take it at the end, right? And then it becomes water. So the interesting thing about this is we always think, the order of breathing is we breathe in oxygen and then something happens inside of our body and we breathe out carbon dioxide, right? That's how we always all intuitively think about it. But now you realize it's actually backwards, right? We release the carbon dioxide first as part of aerobic respiration, and then we breathe in the oxygen so that we have oxygen to kind of finish the step at the end. All right. Neat way of thinking about it. Right. All right. So let's go ahead and now focus on this electron transport system, step 4. Right. A series of big protein complexes that are going to use those electrons being brought to it by these filled electron carrying baskets. And those electrons are going to power this pumping of protons across this inner mitochondrial space. Right? All right, so these are the 3 main complexes of the electron transport system, represented as these big brown blobs here. There are actually 4 brown blobs. There are 4 complexes, Roman numerals 1, 2, 3, and 4. But you can see in this illustration, they just show 1, 3, and 4 over here. That's because the ETS complex 2, right, is usually not associated with this part of the electron transport chain. The complex 2 is actually more associated with the citric acid cycle. So if you go back and look at the citric acid cycle, there's this one step where you're filling an electron or you're transferring electrons out of the intermediate molecules of the citric acid cycle, like the succinate, into an FAD-H basket, right? And so that FAD-H2 basket carrying electrons doesn't really enter at the start of this complex here, right? It actually enters somewhere in the middle. And we'll take a look at that on a later slide. All right. But for right now, to introduce this, I want you to think about this. Each one of these big brown blobs is a whole group of different proteins, right, working together as a big complex, a big holoenzyme or holoprotein type of complex. You know, we've seen that type of thing before, right? What this is showing is if you start from this purple NADH, right, that's carrying a pair of electrons in here, it will empty those electrons into the proteins that are part of this first complex. And you can see that within these complexes are these funny letters, FMN, FES, here's a CYTB, right? Here are other CYTs over here. What are these? Right? What this is meant to show is that these different proteins that make up these big brown blobs, right? Many of them have special chemical groups that are sort of part of them that actually handle the transfer of electrons through the complex. And also there are other molecules like this CYTC here, right, that help transfer the electrons from one big brown blob to the next. Okay. So what are these different types of groups that are actually handling the electrons being transferred? Right. We're going to take a look at those. So what I want you to take home from this slide here, right, is these are the three main ETS complex groups that are associated with the electrons that are being brought in by NADH baskets, right? So these NADH baskets empty the electrons into these different proteins that are part of this big brown blob that each have certain chemical groups part of them that carry and transfer the electron, right? And then it goes through this chain of complexes. As they pass through these complexes of proteins, right, the proteins are changing their shape, right, as these electrons are kind of moving through them, and they evolved so that as they change shape, what's going on? They are moving protons. They are moving protons from the gray side of this slide down here, right? So all these little light blue things are showing you the protons. They're being moved through these complexes against the proton gradient, right, into this intermembrane space of the mitochondria, right, building up many, many more protons on this side. Right. Eventually, step 5 of this that we'll finish talking about today are these ATP synthase complexes that will allow the protons to flow back downhill through them, right? Because that's where the protons want to go, from high to low concentration. And then the movement of this ATP synthase is going to produce ATP. Look here. Here again is where the oxygen fits into the story, right? You can see the oxygen is right at the very end, ready to accept the electrons that are flowing through this big electron transport chain, right? So without the oxygen at the end, what would happen? The electrons would get stuck at this last part here, and everything before it would back up, right? Because if everything gets loaded up with electrons, and nothing is at the end to keep taking these electrons out, this whole thing would come to a grinding stop, right? So that's why, even though, you know, I said maybe it's anticlimactic to realize that this is what the oxygen is doing, right? You realize how important it is, because without the oxygen, this whole system of electron transport and proton pumping wouldn't work, right? And that's why without oxygen, your cells die, right? And if your cells die, you die, right? Okay, let's take a closer look at these funny groups that are part of these big brown blobs here. So these groups are what we call prosthetic groups. You know, prosthetic, you're like, yeah, that's like what? Like a prosthetic means something that you can, you know, like for example, add to your body, like a prosthetic limb, right? So prosthetic groups, the reason they call them that is because these are chemical structures that can be sort of added onto or held onto by certain proteins that can make use of them. I like to think of them as kind of like a baseball glove, right? And how a baseball glove is designed for baseball players, right? It's not really part of the baseball player's body, but baseball players can be equipped with a glove. And the purpose of the glove is it helps really facilitate the catching or the accepting of a baseball and the transfer and the passing on of that baseball, right? In order to have a successful play during a game. Right. So let's think about these prosthetic groups as sort of like these baseball gloves. And of course, the electrons then are the baseball itself. So there are several examples of these types of chemical structures that function as electron carrying prosthetic groups. There are flavoproteins. So there are certain proteins that will have a partial FAD group sort of held onto them. Right. And of course, we already learned the FAD is an electron accepting coenzyme. So if you have part of the structure of the FAD, not the full FAD, but just the part that kind of accepts the electron, if you have a protein that has that, right, it can function as a baseball glove to help transfer electrons around. Not surprisingly, many metal ions are used as part of these prosthetic groups. Things like iron, for example, are very good at receiving and releasing electrons. So some of these have these iron sulfur cores. So you can see here, this iron Fe and sulfur S core sort of forms like this cube. And it is held by the side chains of cysteine residues. So if you have a big protein surrounding this, right, the cysteine amino acid residues in that protein, right, because of the amino acid sequence, they would be positioned in just the right spot where the cysteines would hold on to this prosthetic group. Right? Some of them are cytochromes. So, for example, cytochromes like heme. You can see here, this is the structure of heme. It's a pretty elaborate structure, right? Very obviously, when you look at this, you realize, yeah, this is not a protein. Right? This is just a sort of a standalone chemical structure that has an iron in the middle. Right? And so this heme group is the group that hemoglobin proteins use. So your hemoglobin in your red blood cells, right? The proteins, the hemoglobin proteins, are the right shape to hang on to these heme groups. Right? And that's how electrons get transferred around, how things like oxygen is able to be bound up by your hemoglobin and carried around. And importantly, now you think about this, right? The whole point of a prosthetic group is it makes it easy to transfer things around. That's why oxygen is easily held onto by the red blood cells, the hemoglobin, and it's easily released, right? So cytochromes like heme. And then there are groups like coenzyme Q, which actually don't associate with a protein. So these are cases where you have a baseball glove that is not actually worn by the player. It's a baseball glove that's just kind of floating around by itself, right? But can still function as one of these electron sort of transport molecules. So an example of that is this coenzyme Q, which is also called ubiquinone. So in order for different proteins that are in these big complexes as part of the electron transport system. Right now, hopefully you're beginning to imagine that a big protein complex, a big brown blob, is made up of many of these proteins. And some of these proteins have these different prosthetic groups as part of them. And you can already imagine the electrons being passed from one group and one protein to the next as part of moving through a big complex. Right? And that's what causes the big complex to change its shape in order to start moving protons across the membrane. When you look at this, you imagine, yeah, when electrons are being carried by these different groups, not all of them will hold on to or give up electrons with the same sort of ease, right? Because of the chemical structure here, some of them will want to hang on to electrons tighter, whereas other ones would be more happy just to get rid of electrons, right? Just because of the chemical bonds. What does that mean? If you were to organize then a whole series of proteins through which electrons are going to flow, being passed from baseball glove to baseball glove, you might already imagine the way you would order this is you would have the first baseball gloves at the beginning being the ones that, yeah, they'll take electrons, but they'd much rather pass it on, right? And as you go further along the chain, right, that kind of changes. Then you get to baseball gloves that are more happy taking it and they want to hang on to it rather than give it up. Eventually, the ones at the ends are the ones that would much rather hang on to it rather than give it up, right? And by organizing the proteins that way, you get the flow of electrons to go through. Let's take a look at that. So that's the concept that's represented here in this table. All right. So this is talking about standard redox potentials for these different types of molecules that we've been discussing. Right? What they've ranked these here from top to bottom is that the molecules at the top, right, all of these are electron, transferring types of molecules here. But as we said before, some of them, right, are more greedy and want to hang on to the electrons. Some of them would rather just give them up. So that's how they've ranked them here. For example, NAD+, the empty electron carrying basket, NAD+, right? It'll take electrons, of course, right? Because that's what it's used for. But the truth is, once it has those electrons, it wants to give them up, right? So that's why NAD being converted into NADH, loaded up with those 2 electrons, 2 protons, it's actually less energetically favorable to hang on to those electrons. It would rather just dump them off to the next molecule that comes along that would take them. That's why here's the FAD, right? Another example of an electron carrying basket. When it gets 2 electrons, it becomes FADH2. You can see this suggests that the FAD is more willing to hang on to electrons, right, than the NAD is. Why? Because it's energetically more favorable for it to hang on to those electrons. And so that also means, intuitively, that when it comes time to give up those electrons, because it's not as willing to give them up as the NAD does because of where the electrons are in the structure, the amount of energy that's associated with those electrons that are being transferred is lower, right, than if they were first given up by the NAD. In the middle of this is in bold, is just showing you that if you have 2 protons being the things that are accepting 2 electrons to become a hydrogen atom, where does that fit in, right, in terms of this amount of energy? So they're just calling that 0, right, because that's sort of like what we're using as kind of like the reference point, right, because 2 protons, 2 H pluses getting 2 electrons, becoming an H2, right? If you think about how happy are they to get those electrons versus how happy are they to pass them on, right? We'll just call that 0. You can see some of these examples of things that we've talked about on the previous slide, like the coenzyme Q, the ubiquinone, the free-floating sort of baseball glove, right? It would be here on the chart. Below it would be cytochromes. So cytochromes that have, for example, things like the heme group, that type of thing would be below it. And eventually you end up with oxygen, right? And hopefully this makes sense to you, because we've said that oxygen is a very greedy electron acceptor. It wants electrons badly, and when it has them, it wants to hang on to them, right? That's why they're at the bottom of the list here, because it is more energetically favorable for oxygen to hang on to them, in the formation of water. Right? Another way of thinking about it is, as you go from less to more energetically favorable to hang on to the electrons, as you go from top to bottom, you can also say another way of thinking about it is the affinity. How willing are these molecules to even accept an electron in the first place? And not surprisingly, that would be the opposite, right? Because if something like NADH is wanting to let go of electrons, that also means it is less, has lower affinity to take them in the first place compared to the molecules down the column here. All right, this blank space, what is this? This is just to give you an idea of where some other molecules that we've discussed fit in here, right? Oh, right, so I forgot to mention. So as you're going down from top to bottom, because you're talking about electrons being removed from these molecules and passed on to the next molecule, right? You are oxidizing them, right? So the oxidation of NADH back to NAD+. Right? That oxidation is, of course, linked to the reduction of the next molecule below it on the list, if that molecule then receives the electrons. Right? And in this case, it's because the NADH has a stronger dislike of hanging on to the electrons. Right? And eventually, at the very bottom, the O2 has the strongest like for hanging on to the electrons. Right? So that's why you end up forming water, very stable water. Right? So what is this blank space in here? This is where the oxidation of acetaldehyde into ethanol, right, and of pyruvate into lactate happens. Okay. This is where they fit in. So that's why you can see, remember, these are parts of fermentation. Right? The acetaldehyde not being oxidized, I'm sorry, being further for becoming the more reduced form known as ethanol, the pyruvate being more reduced into lactate, right? I shouldn't have said oxidation, because in this direction, these things are being reduced into ethanol, right? Or reduced into lactate. But remember, as part of fermentation, how do these steps happen? They use the electrons that are coming from the NADH baskets, right? So the pair of electrons that are happily dumped by the NADH, they're dumped onto acetaldehyde or pyruvate to make them reduced into ethanol and lactate, right? And remember, in glycolysis, that's what happens, so that you've freed up the NAD plus basket to go back and help out with glycolysis, right? And so hopefully seeing where these things are positioned on this figure now makes sense to you, right? Because they are lower energy than the NADH is, right? So they will accept the electrons from the NADH. Okay. This figure is another way of thinking about how these different proteins that are part of these electron transport blobs, these complexes, right, are arranged energetically. Okay. I think this is really neat, actually, the way they've done this, because what they've said here is the way they've positioned this drawing is they've put these complexes, complex 1, 3, 4. Now they've also included complex 2, right? Again, this is coming from the citric acid cycle. You're filling electrons as part of FADH, FADH2 that's coming off the citric acid cycle. Those go through complex 2 over here. Each one of these little ovals is an example of a protein within that big blob that's carrying these different prosthetic groups, right? Like these iron sulfur prosthetic groups, these cytochrome prosthetic groups, right? And based upon the energies of where they fit on that previous slide, ranking their relative dislike of holding on to electrons, right, all the way down to ones that really love to hang on to electrons, you can draw it this way. And by drawing it this way, I think this is wonderful, because you can really appreciate, right, when an NADH basket comes with its 2 electrons that it's carrying and it empties into complex 1, right? The red arrow is the flow of those electrons through these different proteins that are having these different baseball gloves, these different prosthetic groups. Ultimately, the electrons end up being received at the very end by oxygen to form water, right? And as I said before, if you don't have this oxygen at the very bottom, everything above it is going to back up, right? Because everything is just going to be stuck, baseball gloves with baseballs in them, right? Not being able to pass them to the next thing in the chain, right? This is also why, if you look here, remember, the 2 electrons that are being carried by the FADH2 represent lower energy than the electrons that are being carried by the NADH basket, right? Again, because of where those electrons are positioned within those nitrogenous bases of these baskets. So that means the 2 electrons only have enough energy to enter the chain here, right? So in other words, the amount of energy represented by these 2 electrons is already low enough that it cannot flow through the chain up here, but it can enter over here, right? Because at this point, this would be coming from high to low, and but it would still contribute, right? Those 2 electrons would still contribute to the pumping of protons by complex 3 and complex 4, right? But those 2 electrons that are coming from the FADH2 basket are not going to contribute to the proton pumping done by complex 1, all right? All right, MCAT question alert, right? Because I've had students who told me, hey, guess what, Dr. Shen, that was on the MCAT, right? All right. So let's talk about this. So these electron transport proteins, oh, and I do want to mention one more thing, right? The way these cartoons draw these big brown blobs, they give you the impression that they're all physically lined up on the membrane, ready to allow the electrons to flow through them, right? That's not true, right? Each one of these big protein complexes, yeah, it's anchored to the mitochondrial membrane, but they're not actually lined up, right? Like a row of chairs or something like that, right? No, they are freely floating around on the surface, and they just happen to bump into each other, right? So really, the way this chain works is as electrons are flowing through each of these complexes, right? If that complex then bumps into the correct next complex, then the electrons go into the next complex, right? And so on and so forth. For convenience sake, we draw it sort of all together like this because we're trying to learn the concept, right? But the reality I want you to think about, right, is it's just all things floating around together, right? Okay. So the proton gradient that ends up being built up by this electron transport chain, right? We said once it builds up in the space over here, this is the inside the mitochondria again, and you have this membrane that divides the inner mitochondrial space from the outer mitochondrial space, and that's where the protons are being pumped, right, into this sort of what we call the intermembrane space, right, between the inner membrane and the outer membrane of the mitochondria. Then those protons are allowed to flow back downhill because they want to so badly, right, through these special ATP synthases. So the ATP synthase proteins, they use this proton gradient made from the electron transport chain in order to turn subunits of the protein and make work happen. So here is the ATP synthase, right? It is an F-type ATP synthase. So you can see here, it has this central core sort of protein, this gamma subunit, and then it has these alpha beta subunits down here. As protons are flowing through this ATP synthase, as represented over here, that causes the shape of this gamma central protein to change. And it basically causes it to rotate in the middle. Right? This is a cross-section over here of the bottom part, showing these beta subunits. So there are 3 beta subunits colored in yellow, orange, and red. Right? These beta subunits are identical to each other, right? But look at how they're positioned. They're positioned surrounding the gamma subunit. So as the gamma subunit in the middle is turning because of protons coming through, it is pushing up against the insides of the beta subunits. And you get an allosteric effect, meaning as the inside of the beta subunit is shoved by the gamma subunit turning around and bumping into it, right? The outer part of the beta subunit changes shape also, right? Because it's a ripple effect. If one side is being disturbed, it's a big polypeptide chain. All the amino acid side chains have to kind of jostle around to get comfortable again. And you end up with an opening on the outside of the beta unit, right? And so this position is called the open position, or O. In this open position, right, that active site of this enzyme can fit an ADP and an inorganic phosphate. And those will both bind in there, okay? And as the gamma subunit keeps turning, you'll see in the loose position here, right, the shape changes again to bring the ADP and the inorganic phosphate together. And then as it turns, right, it becomes the tight position that then actually catalyzes the reaction of adding the phosphate group to the ATP. Right? So because there's 3 different beta subunits, they take turns being in these conformations of open, loose, and tight. And so as a result, with every turn, every single turn of the gamma subunit in the middle, you actually make 3 ATPs that then float off, right? So this is like the crowd going through the turnstile, right? And now you're getting some work out of it. You're using this to make high throughput mass production of ATP. So it's been approximated that the protons that end up being pumped through the ATP synthase due to one NAD basket emptying its pair of electrons, right, translates into 1 full turn of the ATP synthase. So you make 3 ATPs from that. The number of protons that get pumped through this ATP synthase due to 1 FADH2 basket emptying its lower energy electrons is enough to make 2 ATPs. Okay. So this chart here summarizes this here. What we're looking at here is how much ATP you finally get out of the glucose. Right? So on the right is the tally of ATPs. On the very left are the different processes that we've discussed. So the light blue parts are taking place in the cytoplasm of the cell. That's where your anaerobic respiration takes place, right? Your glycolysis of glucose being converted to pyruvate, and the process of enzymes making a couple molecules of ATP through substrate level phosphorylation, right? Hand making them as they work on the glucose, right? Then along the way, the pyruvate being converted into acetyl-CoA, eventually going through the citric acid cycle, right? This is how many electron carrying baskets you're filling, right? And then each of these then, right, will end up powering protons being pumped through the electron transport system as these electrons are emptied from the baskets into those big brown blobs, the complex 1, 2, 3, 4, right, pumping protons, and those protons will then flow through the ATP synthases. And as the ATP synthases are turning, they're producing 3 ATPs per turn. And so that's why you tally it up this way. So the total energy yield from one glucose molecule then, right, with everything added together, glycolysis plus aerobic respiration, so anaerobic plus aerobic respiration, you get 38 ATPs out of one molecule of glucose, right? Instead of just the 2 ATPs that you get if you were to stop at glycolysis, right? So that is a tremendous improvement, and it all has to do with the availability of oxygen. The availability of oxygen to receive those electrons at the end of the electron transport chain to create the proton gradient that can then turn the turnstiles and make lots of ATPs. I mentioned to you that the 2 ATPs that are made from glycolysis, right, anaerobic respiration, only represent about 3% of the potential energy in the glucose, right? And that's very inefficient. Once you add on aerobic respiration, you actually get up to 40% efficiency, all right? That's a big leap from 3% to 40%. 40% of the potential energy in the glucose bonds is converted into ATPs for your cells to use. You might still say, well, 40% doesn't sound that impressive. I thought you were going to say something like 99% or something. 40% is still pretty darn good. Think about it this way. The internal combustion engines in our automobiles, right, our gas burning cars, right, their efficiency maxes out at around 20%. All right. So after 100 years of designing internal combustion engines in cars, right, typically the best they can do is 20% efficiency of converting gasoline into usable energy. So Mother Nature, right, can come up with a 40% efficient mechanism. And that's what powers life. Right? All right. So we're out of time. So if you guys have to go, you can go, but I wanted to just show you this one fun slide at the end here. Okay, so I'll go through this really quickly. You don't have to know this, right? So the moment I say that, like a whole bunch of you log off, right? When you watch like spy movies and things like that, oftentimes like when a spy is like captured, he or she, in the movies, it's very dramatic. They have this like little pill of cyanide or something that they've been wearing in a locket around their neck, and they take this pill right before the interrogators realize what they're doing, and then they manage to actually kill themselves so that they don't divulge the secrets, right? And you often think, wow, cyanide, that's bad stuff, right? How does it actually work? How does it actually kill you? So here is hydrogen cyanide, right? And you might be surprised to know that that's it, right? It's 3 atoms. It's a carbon bound to a nitrogen bound to a hydrogen, right? How could that possibly be deadly and poisonous, right? It's deadly and poisonous because it is the right shape to fit in the cytochrome C. Cytochrome C is one of those important proteins carrying this heme group here, right? So this is a good example. Here's the cytochrome C protein in green. Here is the prosthetic group, the heme group, the baseball glove that it is wearing, right? The cyanide, you can see, binds to the heme group and prevents the heme group from being able to participate in transporting electrons or carrying electrons. So why is that so important? Because the cytochrome C is positioned here in the electron transport system, right? It carries the electrons from complex 3 to complex 4. Without cytochrome C working, the electron transport system stops, right? And so in many ways, it's like removing oxygen from the very end, except for in this case, it happens even earlier in the ETS chain. Right? And so that's why cyanide gas is so deadly, because once your cells have all of their mitochondrial electron transport chains stopped by the disruption of cytochrome C, right, you stop making ATPs, or you stop making enough ATPs to fuel your cell, and your cell dies. So 1 gram of cyanide can kill a 160-pound person in 10 minutes, right? Because the cyanide, because it's so small, it will just diffuse through all of the tissues in the body, right? And get into the cells and kill the cells because they shut down the aerobic respiration. All right, let's go ahead and stop there. I said it was a fun slide. I don't know what's so fun about teaching you guys about how cyanide gas kills people, but you know, whatever, right? Okay. Okay, let me stop the sharing here. And I'm going to stop the recording.